What would you do if a DNA test proved that you are not the biological mother of your own children? This is exactly what happened to 52-year-old Karen Keegan from Boston, and the truth behind it stunned the entire medical world. A routine test became a genetic mystery. Karen needed a kidney transplant, so doctors tested her 3 adult sons to see who could be a possible donor. The lab results confirmed that all 3 were definitely brothers, but they also stated that 2 of them were not her biological children. At first, doctors assumed there was a lab error and repeated the tests, but the results came back exactly the same. Karen Keegan and her sons received the confusing DNA test results from a doctor. On paper, DNA was claiming that this mother was not the biological parent of the 2 sons she had raised. The shocking answer? She was her own twin. A specialist team at Boston's Beth Israel Deaconess Medical Center decided not to blindly trust the DNA results. Instead, they investigated deeper. They collected samples from multiple parts of Karen's body: blood, cheek swabs, hair, and preserved tissue from earlier surgeries. Scientists analyzed various samples from Karen, discovering 2 distinct DNA profiles. What they discovered was incredibly strange. The DNA in her blood showed one genetic profile, while the DNA in her tissue showed a completely different genetic profile. And that second profile matched her non-matching sons. Karen was diagnosed as a Tetragametic Chimera. In simple words, she had absorbed her own twin in the womb and carried 2 different sets of DNA inside one body. An illustration of Tetragametic Chimerism, showing how 2 embryos can merge to create one person with 2 DNA sets. 1 woman, 2 genomes, 4 HLA types. In some tissues, doctors found 4 different HLA types, immune system markers. 2 belong to one genetic line and 2 belong to another. One DNA line matched her sons and brothers, while the other matched only her bloodstream. Her eggs came from one genetic line, but most of her blood came from the other, which is why standard blood-based DNA tests were picking up the DNA of her internal twin, not her own. There were no outward signs of chimerism. For 52 years, she lived like any normal human, unaware that she carried 2 genomes. How Karen Keegan's case saved another mother. In the same year, 2002, when Karen's case was published in the New England Journal of Medicine, another woman, Lydia Fairchild, was told by a U.S. court that DNA proved she wasn't the mother of her own children. Fairchild was accused of fraud and faced losing custody of her kids until her lawyer discovered Karen Keegan's published case and introduced the possibility of chimerism. A deeper test using a cervical swab finally revealed a second DNA line in Fairchild, one that matched her children. Karen's case became the key scientific evidence that proved Lydia was indeed the biological mother. Why this story matters more than you think? Karen Keegan's case forced scientists, doctors, and even courts to acknowledge a difficult truth. DNA is powerful, but not perfect. In extremely rare cases of chimerism, blood or saliva DNA tests can completely misidentify biological relationships, leading to life-changing legal and medical consequences. Today, her story is taught as a major lesson in genetics, law, and forensic science. Before destroying lives based on one DNA report, remember how strange and unpredictable the human body can be.